Hello, I'm Katrin. I'm also from the detection dog team. I am from Germany, so please excuse if there's a word or two that I don't remember. And I'm going to talk to you today about the koala genetic research that we've been doing at UC in combination with the detection dogs. And our team, Roman briefly mentioned it, but I just wanted to introduce the team a little bit more. The detection dogs team has been established in 2015. We have currently 5 specially trained detection dogs, mostly for koalas, or they're all trained for koalas, some on scats, some on living koalas. Well, we also have 16 humans in total, so that's a lot of them students and a couple of people who help us deal with all the other stuff. And so far as a team, we have done over 4,200 surveys across Southeast Queensland and New South Wales in those, 7 years only. And 950 of them roughly have been done in Redlands, Mainland and Minjerribah. And we have so far collected roughly 2,900 koala scats for genetic analysis. So that's quite a lot of information that we are processing. So 2018, this is me sitting in William Ross Park, I think it was, with my detection dog, Billie, that I've been working with, trying to understand how the Redlands is looking, where all the parks are, where all the koalas are. And this is where it all began after Kath reached out to us and we started doing our koala monitoring. And basically what we did is just go out in the field, look for koala poo, and collect it all to analyze it and look for genetic information. And there I just want to give a very brief overview without boring you to death about the specifics, just to understand why it's important that we actually look at these things. So conservation genetics is the combination of conservation biology and genetics. And there's a lot in genetics that we can find out. Basically, when we talk about genetics, we talk about the DNA, and the DNA kind of works like a computer. Computer background is set up from zeros and ones. Everything is zeros and ones, and the DNA is everything is TCGNA. So they're just 4 letters, and they make up everything. And we have basically a little computer in each and every single cell of our body. And there are some variations to so every gene has a combination of these letters. And the slight differences in these combinations is what makes us, or what gives species the ability, or populations the ability to adapt and change and be flexible, and that's evolution. And so basically, yeah, this diversity in the sequences is what we call genetic diversity. And genetic diversity is one of the most important things that we look at in conservation genetics, because it tells us a lot about how the population is faring. And basically, to put it very simple, is high genetic diversity means that we have a lot of these variants, and that means that there's a higher chance that if something changes in the environment, such as climate change, there might be a couple of koalas in the population that have a specific variant that can handle heat better, or that can handle thirst better. And that means that even if the rest of the population dies, there will be a part of the population that survives, and overall, the species can survive, and the population. And conversely, adversely, low genetic diversity means that there's very little flexibility to deal with these future challenges, and there's a very low chance of survival in case something, or like a new disease is introduced, for instance. So yeah, there's extra hidden information that we can't see by just looking at the koalas. And this is what we can extract from the DNA from those 4 letters, which is amazing. And what I just talked about, the genetic diversity, translates into the adaptive potential of a population and of a species. But there are other things, such as historic and contemporary population dynamics. So something we look at is sex ratio, for instance, which obviously, using genetic data from poo, we don't actually have to find every single koala in the population, but we can get a pretty good sample size just from collecting the poo. And so we can tell about sex ratio. We can see how the koalas are communicating across the landscape. Is there gene flow? Do koalas disperse, or are they kind of stuck in their suburb? And we can also see whether there are areas that are isolated. And therefore, we can come to some conclusions about the population viability in general. But also what's really interesting is relationships. How related are the koalas in the landscape? Do we have Mom and Joey sitting close to each other for a long time, or do we actually see them move around the landscape, which also gives an indication of fragmentation and how it affects the population. And then obviously an important one is disease. We do have in our data that is like a genetic capture mechanism, something that can capture DNA fragments from chlamydia. And so we can tell from any poo sample that we collect whether that koala had chlamydia or not, which is very important because sometimes the presence of a chlamydia in the poo can tell us whether this pathogen is present, which doesn't necessarily mean that the koala has clinical signs of chlamydia. So in 2018, we did our first round of genetic sampling, that is poo sampling for genetic analyses in wetlands. And this is all part of a genetic monitoring program. So we went out again in 2020 and a little bit in 2021 because we had some COVID-related delays. And we will go out again this year. And we hope to continue this monitoring. Genetic monitoring is something that's being more frequently used all across threatened species, vulnerable species, but also common species, because it can tell us a lot about the population. We can identify unique individuals, and sometimes we can recapture the same individual 2 years later, but we also find a whole heap of new individuals, which is pretty cool. And we can quantify changes, which is really important. So we can see, we measure a lot of genetic matrices, and we can see whether they change. So for instance, the sex ratio, does it change? The genetic diversity has a whole heap of different measures that we can do. Does that change? Does it stabilize, or where is it going? And we can track the changes of the individual distribution over time too. So because we try to cover the whole of the wetlands, we can see, are they always at the same spot? Sorry, I'm really cold. I'm not shivering with nervousness. I'm just like really cold. So to explain this just very briefly, so we have time point zero. So this is genetic monitoring in pictures. We have time point zero. We go out the first time. We have no idea what's really around. We have a look. Okay, this is kind of what we think. We go out again time one, and we find a heap less. This is just very randomly to explain it. This is not the redlands. And then what I want to point out here is that you have time zero and time one. Between 2 points, you can draw a straight line. So just having 2 points, 2 measurements, we can't really tell anything. Is it variance? Is it completely random? Or is it actually a trend? It becomes important that we keep monitoring and we keep collecting this data, while we also, of course, make recommendations every year on the things that we do observe at that time. And then hopefully at some point in the future, at time point 2, 3, 4, whatever, we see that our measurements that are being put in place are actually working. And this is where we need to monitor the population. So what I did in 2018 is I went out with my favorite of the dogs. That's Billie Jean. She's about 8 years old now. You can see that during the time she's got a lot more gray in her face. She's trained on fresh scats only, which is really important because there's obviously a lot of old poo in the redlands, but we really only want the fresh poo because that's where the genetic data that we get from the DNA is the best. It kind of degrades over time a bit, which also means that we can't use all the samples that we collect because some of them are just not good enough, and that could introduce some false data. So we always only use a selection of the samples that we actually collect. And Billie has done about 800 surveys, so she's quite experienced. And we went out in the field, and here you can see the dog track. So the dogs wear a GPS collar, and with that we can track exactly, this is where we started, this is where we went, this is where we looked. It's still just a line on the map, but really the dogs we don't know exactly, but they have kind of a cone of sense, smell. So it's not that they just smell that one line, but they can kind of direct where their scent carries them. Smell carries them. And you can see that we try to cover everything from top to bottom. And in 2018, it was kind of the first time we did this. So we then went and also had a look of where we had gaps in the landscape, where should we have a bit more effort. And we went back in 2020 and tried to fill some of the gaps, which sometimes worked, sometimes didn't work. But at least we were able to repeat all of the surveys that we've done in 2020. Here you can see the positive and negative survey sites. So survey sites that we've visited and repeatedly, and when they were positive for koala signs, that is poo, and when they were negative. And you can see here that there are some areas where we constantly find negative results and some where they're always positive, but then also some where it changes. So for instance, Thorn side, we haven't picked up any fresh koala poo over the 2 years, but then for instance in Venman National Park, we had fresh poo in 2018 and then not in 2020. Again, this is only 2 time points, so we don't know, is that just a random variation, the koalas are just moving around somewhere, or is this actually a trend? And the other thing that we have to keep in mind is that every time we go out with the dog, we spend 1 or 2 days in Thornside, a couple of hours a day. That doesn't mean that koalas are not there at all. That just means that at that point when we looked, there was no fresh poo present. There might have been a really old poo that the fresh scat detection dog couldn't pick up, but it definitely gives us an idea of how frequent koalas are in that area, how often they are encountered. Doesn't mean they're not ever there. And to touch on what we did with that data that we collected, so at the positive sites we collected poo, and Roman showed you earlier how the dog actually finds it. We then bag it in a little tube, and what we do in the lab is just very briefly, we scrape the outer layer. The idea is that the poo travels through the koala and on the way through the intestines collects DNA cells from the koala's intestines on the outside. We then scrape the outside because we don't want any of the other stuff, for this study at least, where it's bacteria and plants. And then we wash it, we isolate the DNA, and we send it off to experts to give us the correct genetic data. To recap our efforts in the Redlands, in 2018 we have done 228 surveys across the landscape. We collected 236 genetic samples. That is only the mainland, not including the Manjeraba data, where it was probably just about the same, if not a bit more. And from the genetic samples, we identified 124 unique individuals. So you can see how it's less than half, or a bit more than half of the genetic samples that we collected. So part of that were duplicate collections, where we collected the same individual multiple times, because you can't tell from the poo if it's the same koala or not. And some of that is just data not being good enough. But our technology also constantly improves, and we are able to retain more and more samples. And then in 2020, we did a bit more in the surveys, trying to cover some of those gaps that we identified. And Kayo was there in the field. You probably have, some of you might have seen us knocking on your door, Kayo or me, over the last few years. And we collected just about as much samples and identified 116 unique individuals. That does not mean that the koala numbers dropped. That's just a momentary number that we got out of the genetic data. What we get, what I mentioned earlier, is, for instance, sex ratio. Sex ratio is really important because we want to have a balanced sex ratio in the population to be sure that they're able to breed and produce offspring. Ideally, we would have slightly more females than males, but the balance should be quite balanced. And we can see that in 2018 we had slightly more females, and in 2020 we had just a few less females than males. Again, this is not a trend, this is just 2 points. So again, making the point that this needs to be monitored. It's definitely something to keep an eye on. So if we have a result like this where we see, okay, for some reason we now have less females, is there something that we have to keep an eye on? So this year we will have a very, very specific look at the sex ratio and see if the number of females have dropped more, because that could obviously be a problem for the breeding in the population. The chlamydia results, this is going hand in hand with the results that we have from clinical assessments and also from what we generally observe in the population, that about a third of the koalas that we sample have the pathogen present. And this has slightly increased between the 2 years, but the increase is very small. So yeah, we will see how that develops, but with all the measurements that are going on and that we are trying to implement, we hope that this number is going down. And what we also looked at is some of the more deeper genetics. So we found that overall the population is one breeding population. So they're not massively fragmented. They're still communicating with each other. They're still dispersing in between. But we found some more cryptic shifts where the koalas in the north seem to be more similar to each other and the koalas in the south seem to be more similar to each other. This is a very cryptic, like not very strong signature, but it's definitely also something to keep an eye on. It has been consistent across the years. It does coincide with some of the more major infrastructure in that area and also increase in traffic in that area. So it's definitely something to keep an eye on, that we don't isolate those two parts of the redlands. Skip this, yes. And these are the genetic matrices and the measures of heterozygosity that I mentioned earlier. HE, HO, it doesn't really matter what it is. It's heterozygosity, which is one of the many measures for genetic diversity. And basically, the higher this number is, the better. The number alone necessarily doesn't mean anything unless you compare to something, either a different time point or another population that has been assessed with similar methods. And what we can see is that the genetic diversity just slightly, I can't point it out, but it slightly decreases from 2018 to 2020. And the FIS measure is a measure of inbreeding in the population, and it slightly goes up. So it is based on the measures of heterozygosity, which is used to calculate inbreeding. So we have a slight increase here. We will see in the next round if this is actually ongoing. What we have also done, which is actually a third measure, but it's quite far back in the past. Well, not that far. We got 261 tissue samples from the redlands that were collected from wildlife carers, from hospitals by Deidre, and we're very thankful that she donated these samples to us for our research purposes. So we had a third time point from the past. And we can see here that the change in those 12 years is actually quite dramatic in comparison to the last 2 years. So we see that heterozygosity was much higher then, and we see that inbreeding was much lower then. And one thing I should probably mention here is that these particular measures of genetic diversity, they have some sort of time lag to them. So you carry the genetics with you, you inherit them from your parents and your grandparents and so on and so on. So what we see here with these measures can be time lagged. It can reflect more on the measures from the past few generations. And there's one measure that kind of reflects more contemporary situations, genetically speaking, and that is, for instance, relatedness. So relatedness is contemporary because you're brother and sister now and not in the past. And we can see that the relatedness, the general relatedness amongst koalas in the redlands has increased on average, but also that there are now pockets that we found where koalas are closer together, where they're more related to each other than they necessarily should be. Again, we will keep monitoring this. This is also just because we collect the samples over the period of 2 to 3 months normally. It could be that there are some joys present or that there's something going on that will then go away after. And so we'll see how that looks this year. And to recap, what we know is that the redlands and overall koala coast and overall Southeast Queensland koalas have been in decline for the past few decades. And we can see that in the genetic data. We have a loss of genetic diversity and we have an increase in inbreeding generally. We know that habitat reduction, so land clearing and other things, and fragmentation are ongoing. We know that the human population in redlands has grown by something like 16% over the last 20 years. and that chlamydia remains prevalent. So that's not going anywhere. It's been here for a while and it's not getting any better. This problem of the overall decline is not necessarily a problem unique to the redlands. We've already established that. But it has reduced the gene pool of the koalas, of the koala population, has reduced the genetic diversity, therefore has reduced the potential of this particular population to deal with any ongoing or, new incoming threats, whether that's a disease or a change in environment or environmental catastrophes. And we have seen that, unfortunately, inbreeding has increased, especially when we look at the time point back in 2006 and compare it to 2018. And that the relatedness amongst them has increased, which could also be because there are a lot less koalas now. And that there's some very cryptic substructure in the population that we just need to keep an eye on and maybe investigate further what the reasons for that could be. And the future outlook, I was struggling with this a bit. What can we do with this information? Because basically Redlands City Council is already doing so much. And every year with our report, we give recommendations and we find that they are all implemented in the next period. So basically what we really need to do to maintain, if not increase, the genetic diversity and therefore the future viability of the koala population from a genetic standpoint is really just improve the numbers, increase them, or at least keep them stable. And just by increasing the population size, there will be more natural genetic variation and more options to breed with, I guess, and less relatives to breed with. And making the road safe, citizen science is a really important one, so we can keep track of them. Maintaining habitat and habitat connectivity, which there are also plans for. Maintain the monitoring, that is the monitoring of the koalas, but also the genetic monitoring so we can get that third time point and hopefully the fourth and fifth, and hopefully see the trend go up. And what I also really want to point out is the education. My passion is educating kids and go to schools and give talks at schools. And if you have that opportunity in one way or the other, that's also really important to reach out to the young ones. And they're normally really interested and really love it. So that is me. Thank you so much for your attention. And I'm really looking forward to your questions later. And it's great to have you all here.